So in this study, we did animal experiments and basically tested a wide variety of cancers, some of them human cancers and some of them mouse cancers. And these range from breast cancer to ovarian cancer to melanoma, glioma, and other types of cancers. And what we found was we had previously found that fasting and fasting cycles could protect the mice from chemotherapy side effects, but it was not clear whether this effect also applied to cancer cells. So if we protected the host by also protecting the cancer cells, the study would be worthless. So we spent basically 5 years actually, working on the cancer. What happens to the cancer cells? And we showed that the fasting cycles make it a lot worse for the cancer cells when chemotherapy is provided. And even more surprisingly, without chemotherapy, in a number of different cancers, we saw that the fasting cycles were as effective, or almost as effective as chemotherapy. This did not happen all the time, but it happened in multiple types of cancers. And so those are, of course, very exciting results for us. Yeah, so the focus, it's been on the magic bullet, how do you kill a cancer cell, primarily. And so what this paper showed, that in addition to going after the cancer by just protecting the normal cells, here we're also showing that maybe one way to go after the cancer cells specifically is not by figuring out or finding a magic bullet, but by creating extreme environments that a normal cell can respond to and a cancer cell is unable to respond to. And so with each mutation, with each oncogenic mutation or tumor suppressor mutations that a tumor acquires, the tumor has to give up something. And we think that something is the ability to deal with extreme environments. Fasting is probably the most extreme, or certainly one of the most extreme environments that you can encounter, and that's why we think the cancer cells have such a difficult time dealing with it, because there are major reduction in glucose, major reduction in growth factors, and so many things outside of the cell change that the cancer cell is no longer able to deal with it.
To understand the mechanisms by which fasting protects healthy cells, Longo and colleagues analyzed the changes in gene expression, regulating the normal function of the cell. Therefore, they selected cells from the liver, heart, and muscle of mice that were either fed a normal diet or from mice that were fasted for 48 hours. After 2 days of fasting, Longo detected a drastic change in the expression pattern of these genes, which modify the cell function and reduce proliferation while putting them in a protected mode. Healthy cells enter this protective mode to survive times of low or absent nutrients as present under fasting conditions. But if fasting protected these healthy cells, wouldn't it also protect cancer cells? When Longo compared the normal and healthy cells to cancer cells, 2 days of fasting caused a gene expression pattern in cancer cells opposite to the ones detected in normal cells. Because cancer cells have undergone genetic mutations that cause a growth advantage under normal conditions, they have lost the ability to deal with stress caused by fasting. So does fasting have an application for cancer patients?
Yes, there are a potential application for cancer patients, and we already published a few years ago in connection with our protective effects of fasting. We showed in a case report of 10 patients. These patients basically did receive chemotherapy with or without fasting, and they reported that they did much better or better when they fasted in combination with chemotherapy.
When we heard Dr. Longo's findings with fasting protecting against chemotherapy toxicity in mice, we were really excited to move the concept into people because we felt that it could have broad applications and it was free. The other thing that was really unique about it is, unlike very specific targeted pharmaceuticals, fasting could have a wide range of effects on both cancer tissue and normal host tissue. and making it really attractive and potentially very powerful. So we started a phase 1 trial. My role has been principal investigator, and we had 3 cohorts planned of fasting, 24, 48, and 72 hours. The phase 1 trial is about 80% complete, and our Data Safety Monitoring Board have met after each of the cohorts have been filled and have allowed us to escalate to the next dose cohort. We're looking forward to presenting some of our results at the ASCO annual meeting in June.
We think because the results are so remarkable, we think that oncologists who are in fact in charge of the diet of their cancer patient should consider for a particular type of patient who may not have other options, should consider the fasting right now and determine whether, in their opinion, the preclinical data and the preliminary clinical data that is available is sufficient to make the decision that they should fast in combination with chemotherapy. So we don't suggest that the patient goes out and does this on their own, because there are potential problems associated with it, like reduction in blood pressure, and for patients, for example, that lost a lot of weight, this could be very problematic. So the oncologist needs to be in charge, or a physician needs to be in charge, but together with the physician, they can make the determination of whether this is something that they want to attempt even before the clinical trial is completed. Now, keep in mind that to investigate clinically every chemo with every different type of cancer, it could take 20 years. So it's unrealistic to expect that the oncologist will have to wait for their particular type of cancer to make this decision on whether to take advantage of this intervention or not.